this method is already available, and this is also something which uses nanotechnology, biotechnology, and so on. While neither of this technique nor viral, non-viral is not ideal, some hybrid methods also exist. They combine two or more techniques. For example, virusomes combine liposomes with an active HIV or influenza virus, mixing viral vectors with cationic lipids, hybridizing viruses, and also viral pseudotyping when the non-specific protein receptors are incorporated into certain virus. A very interesting approach. Here we see, for example, two viruses. And after the pseudotyping, we see that the blue one is covered with the envelope of the red one. Yes, and here we see that mixture, viruses recombination, when the genetic material of two different viruses is mixed one with another. Why it is important? For example, we have virus of C, hepatitis of type C. It is dangerous or not? C hepatitis virus, is it dangerous? What do you think? Dangerous, of course. But for millions of years of its evolution, it is very good for penetrating the liver cells. And we have the boy with the absence of the factor 8, which is responsible for blood clot. And this guy has low blood clotting, and we should deliver the gene into the liver cell. And usually the receptors, which are responsible for penetrance of any virus to some cell, for example, they are always on the surface of the virus. So we can take the surface, just surface of the C hepatitis virus, and envelop it by the surface. Another virus which is able to transfer the factor 8 into the liver cells of our body, for example. In such approach, we receive a molecule which is recognized by the liver cells. The liver cells think that this is C virus and let enter it into the liver. And however, it is not dangerous for us, and it is able to transfer the gene of our interest into the liver. That's why this pseudotyping, to some extent, is also very useful. And gene therapy, generally divided in two categories. These are approaches how we deliver the gene. In ex vivo gene therapy, and you should write please, two categories of gene therapy. It is important. If you haven't written, for example, the previous material, some of you did but not all, this one is mandatory to be written. Ex vivo gene therapy, human cells are taken out, transduced with the target gene, and finally transplanted. In the in vivo gene therapy, the gene itself is administered into the human body directly. So we are speaking about two main approaches of gene therapy, ex vivo and in vivo. Ex vivo gene therapy and in vivo gene therapy. Now we will see picture, but at first let's imagine that cells are taken out, then transduced, changed with target gene, their number is increased, and then they finally transplanted back to the patient's body. And in vivo, when gene of interest is delivered directly into the human cell, into the human organism. Ready? Steady, go. For the next picture, of course. It's okay. It's okay? Okay. So let's go. So this is the picture to imagine what you have just read. Direct delivery of a gene when we have treated virus and we deliver it directly to the liver. How does it work there? We can know more or less because we must be sure that we are able to deliver this gene of interest into the liver. And cell-based delivery, when the therapeutic tissues are taken out from the human body, it is some vectors delivered there, and they must transmit the gene of interest into the cell in culture, and then these cells are again checked. The number of them is restricted to those which had taken the gene of our interest, and only after that, for example, we increase the number of the cells and then we put them back, incorporate them into the human body. And this is our picture, which is well known to us. Tell me, people, please, what type of gene therapy is used here? You have just written that, so please tell me which type of gene therapy do we see? Are we seeing now? In vivo gene therapy. Gene therapy in vivo, this is the right answer. Because then we put the virus, which brings our gene of interest directly into the tissue. That's why, of course, this is in vivo gene therapy. Because it is the gene of interest incorporated like this, and it must work in the tissue of interest, because this is vector by itself. The vector by itself is brought into the human body. By the time of application in ontogenesis, gene therapy is, you are right, fetal gene therapy of fetus. Human civilization, medicine, including Ukraine, also is able to change some genes, genetic approaches, genetic programs of the fetal tissues or fetal organism, which is not born yet.
Exactly. Someone won't again or...
If, for example, during the testing, screening of a pregnant person, we can see that something is wrong, and this complaint is under the responsibility of gene therapy. For example, we can treat this complaint before the birth of the child. And in this case, we apply fetal gene therapy. It is also done in Ukraine. In Kharkov, for example, in Kiev, there are two institutes which are responsible for this work. Somatic gene therapy of body cells of certain patient. This is when the person, what we have spoken about, when the person is adult, when we see some problem in this person, and we can treat this problem while using the gene therapy technique. Perspectives of gene therapy. Gene vaccines can be used. Gene therapy can be used, and it is in oncology. But this is three perspectives. This is also the highest aim, and it is in your test, in your final test. I input this into your final test, so you should understand it from now and then, that the main goal, the main perspective of gene therapy is providing a single treatment are enough for having constant therapeutic effect of patient's disease. Usually, as we have spoken, for example, for adenovirus, which is spread in the nasal passages, this gene therapy are short-living. It lives for 2, 3, 4 weeks until the tissues of nasal mucous membrane are replaced. And after that, the treatment must be again repeated. However, maybe fetal gene therapy can bring us to this solution. Then the treatment is provided just once, and it's enough for the whole life of the patient. But can you tell me, do you imagine, are these people who are genetically treated by gene therapy, what genes would they pass to their offspring? What are the ideas? which genes could be transferred to the offsprings?
Expect me would be.
I think this artificial genes, the one like
Could they be transported to the offsprings, transferred to them, inherited? Only in one case. What is this case? When these genes would be inherited. Only in one case. What every of you gives to your child will give to your future child. Hmm? What would you give to your children in future?
Genes.
Genes, exactly, in form of what? Okay, where are they? Where these chromosomes are enclosed in what? They are enclosed in something. What are you going to give to your children? Hmm? Samia, what are you going to give to children? Okay, egg cell and sperm cell. Am I right? Some of you will give to your children egg cells, and some of you sperm cells. And the gene of interest, for example, the only one case to give to the children normal gene if it is already somehow inserted into the cells of sex cells. But if we apply, for example, somatic body gene therapy, neither of this gene nor their products will be in the sperm or egg cells. That's why, unfortunately, children inherit the genes of the body, which it's also in turn inherited from its parents, from grandparents. And we are speaking about genotherapy as a promising approach, nice approach, and we maybe would can have it with the more spread technique. However, we have a few shortcomings of genotherapy which prevent it from becoming effective treatment for genetic diseases. It is short-lived nature of genotherapy. This is immune response of the human body to this new incorporated genes, and problems with viral vectors. We see that they are harmful, and it's not always possible to reduce this effect. And multi-gene disorders, usually by means of gene therapy, can be treated only the disorder which is based on the problem with only one gene. Monogenic but not multigenic disorder. And insertional mutagenesis is the illustration to those which we just now have seen. This is mutation which is caused by insertion of new genetic material into normal gene. For example, we cannot control it always. If we put our virus with desired gene into the body, we cannot be sure always that it will not come and disintegrate coding region of the gene. The new portion of DNA you wanted to be inserted into the human genome can come and cut coding region of normal working gene. So it can lead to cancer, for example, as we have spoken on the previous lecture, when the problems with control system of the gene of the cell cycle can lead to mutagenesis, can lead to cancer. And the next approach of usage of recombinant DNA technology, of course, is transgenic animals. And they are truly animals with the genes of, it is, you see, it's taken, the picture taken in dark, and this is the, you see transgenic animals with, again, some fish genes which are responsible for fluorescence, and you see that it is very nice. And we will continue after the break with you, so you are welcome to be back for the second period, and because there are a lot of interesting pictures in the our future in future slides. Thank you for your watching.